Okay, how can you have situations of non-dominance? So we start looking at this population of flowers, 4 o'clock flowers. And so you have 2 pure breeding populations of these flowers. One have red color and one have white color. So again, we know that a pure breeding plant is homozygous, so they cannot be heterozygous. And so we give these allele names, but we're not going to follow the standard way we would do this. So we might have done it before, like big R, little r. So if they had homozygous red, they were red. If they were little r, they'd be white. And so the reason we don't do that is because this implies dominance recessiveness. So this implies that the big R is dominant to the little r. So instead, we're going to use a different naming system. And so we're going to name the alleles CR and CW. And so again, the flowers are diploid, so they carry 2 alleles and they breed true, so they're homozygous. So this red flower has both alleles that are CR, and this white flower has both alleles that are CW. So what happens if we cross them? So we know that this can only produce one type of gamete, CR. We know that this white flower can only produce one type of gamete, CW. And so when we combine these together, we get a heterozygote flower that has one copy of each allele. And so these flowers, they almost follow that old blending inheritance that we talked about. And so the heterozygotes no longer have the same phenotype as one of the parents. They're not red, they're not white. And so in this case, these flowers are pink. So these pink flowers, this is the first kind of surprise. This is the first indication that it's not going to follow Mendelian genetics because it's a brand new trait. So if we continue to do this cross and we do CDAR, CW, we know that this individual, this F1 individual, can actually produce 2 different gametes. So it can either produce a CR or a CW. And so if we simply follow Punnett square, we can produce this Punnett square. So the sperm can either have one or the other. The egg can either have one or the other when we do an intercross. And so if we fill out this square, we find that there's a 1/4 possibility of having this, a 1/2 possibility of having this, and then a 1/4 possibility of having this. And so this is our second surprise. It results in traits with a 1 to 2 to 1 ratio in F2s. So if you see something like this, so if I describe a cross and I say the F1s had a brand new phenotype, and then in the F2s they followed a 1 to 2 to 1 ratio, immediately you should assume, okay, so maybe recessive dominance isn't the case. So we give names to these situations. So in the first case where we have pink flowers, we call that incomplete dominance. And so incomplete dominance is kind of like a blending between the 2 traits. So if red was dominant to white, if the interaction was dominant, then we would see the heterozygotes would be red. If it was recessive, we'd see the heterozygotes be white. If we see it to be an intermediate between the 2, we can call it incomplete dominance. And then there's also situations where you see both of the parental phenotypes. And so both of the parental phenotypes, we see both the red color and the white color. And so that is what we would call codominant. So this is just an example of how you can have different types of allelic interactions than you might think. So typically we use symbols for alleles, and we try to use alleles that allele names that kind of convey the type of interaction that they have. So if there is an uppercase letter, capital D, that is matched with a recessive allele, little d, then very often what we're trying to indicate is that this recessive dominance relationship occurs. Other researchers that study model organisms often use different symbols for alleles. So an example of this is actually in Drosophila. So Drosophila, they do not follow this convention, and instead they typically think about things as either wild type or mutant alleles. And so the wild type allele is what a normal fly in the world would have, and because they do a lot of experiments where they actually mutate the flies, they consider the alleles that they study as mutant alleles that occurred in the lab. And so body color is an example of a trait that researchers have studied using this fruit fly Drosophila melanogaster. And so the mutant phenotype is, the mutant allele is shown by a little e, and then the normal wild type allele is either shown with an E plus or just a plus. And so these are different ways to show the same genotypes. So in this case, we call the wild type, homozygotes for the wild type can either be E plus or E plus, and then another way to do it is plus plus. So just be I don't want you to have to really memorize the rules of allele names yet, but we start to see that there's a lot of different times. So allele names no longer follow these set rules, and so you're going to have to start to get used to being able to kind of get used to the fact that there's a lot of different naming conventions for how to name alleles. All right, so you can also have multiple alleles segregate in a population. So in here, there was still only two alleles. There was a red allele, there was a white allele, but we now saw a third color just because the heterozygotes had a different phenotype. But you can also have situations where you have more than two alleles of a gene. So here is a case in rabbit fur. So the color of the fur of rabbits that we might be interested in. And so researchers have shown that there are 4 different colors that the rabbits can have. They can have this wild type brown fur, so the rabbits are all brown. They can be this chinchilla color, so they are black tipped white fur, and so it kind of gives them this black and white gray appearance. They can be Himalayan, which is white fur, with the exception of certain patches on their paws, nose, ears, and tails. So they're largely white, but also have black on it. And then they can be albino, where the rabbit is completely white, and then the eyes are pink as well. And so the genetic basis of this was figured out because they figured out that there's actually 4 different alleles that are segregating in the population. And so if you're homozygote for this wild type C allele, you will have brown fur. If you're homozygote for the chinchilla allele, you'll be chinchilla. If you're homozygote for the Himalayan, you'll be Himalayan. And if you're homozygote for the albino little c allele, then you'll be albino. So you'll notice that we've just listed the purebred alleles, the purebred genotypes, and what their traits are. So researchers have also done crosses. So what happens, for example, if you cross a wild type brown rabbit, a true breeding brown rabbit, to a true breeding chinchilla rabbit, they also found that there were dominance and recessive relationships involved as well. So if a rabbit had the C plus allele, they were always brown. And so this is either the true breeding homozygote, or if it carried a chinchilla allele, a Himalayan allele, or an albino allele. No matter what, the brown allele was dominant over these other 3. For the chinchilla was recessive to the brown, but it was dominant to the Himalayan and the albino. And so that means not only the pure breeding one, but also any of the genotypes that carry the Himalayan or the albino also were chinchilla colored. And then finally, Himalayan, that was recessive to chinchilla and wild type, but it was dominant to albino. So these 2 genotypes could result in a Himalayan color. And then the albino, the only way the albino could be albino colored was if it's homozygous for both of these alleles. So this is an example where you have 4 alleles, but again, these alleles now just have standard kind of like recessive dominance relationships between them. And so one thing that's important to remember is that when we talk about recessive and dominance, it's always a pairwise interaction. We're always talking about comparing 2 different alleles together. So, let's say we took a cross between the chinchilla and the Himalayan rabbits. So the chinchilla rabbits, and they're pure breeding. So we know that this rabbit can only produce 1 gamete, and this rabbit can only produce 1 gamete. So the F1 individuals would all be heterozygote, and they would all be 100% chinchilla, because we know chinchilla is dominant over Himalayan. What if we did an intercross? So now we have to do a Punnett square. And so in the Punnett square, we now have these possibilities. So, we can fill this all out. And so the result of this would be that you'd have 1 square that was homozygous chinchilla, 2 squares that were heterozygous, and then 1 square that was homozygous for the Himalayan. So in the F2, you would see a 3-to-1 ratio. So again, this in a lot of ways just looks like a, and I guess I filled it out right here, sorry. So in a lot of ways, it looks just like a monohybrid cross. But we can also, for example, do a cross between heterozygote animals. So if we took a chinchilla animal that had this genotype right here, so it had the chinchilla allele and the Himalayan allele, and we crossed it to a Himalayan animal that was Himalayan and also had an albino, then the F1, these animals would all be 50% and 50%. So 50% would be chinchilla and 50% would be Himalayan. And so you can figure that out just by doing this square kind of right here. One final real-life example of multiple alleles are blood groups. So human ABO blood groups, you're either type A, type B, or type O. And blood group, for example, has been implicated as a risk factor for COVID. It's also very important for whether or not what blood you can receive or what blood you can donate. And the idea is that there are antigens, A and B antigens, that present on the surface of red blood cells. And there is a single gene that is responsible for what you present, but it has 3 different alleles that we have to worry about. So the phenotype, so we have 4 different blood types. So you can either be A, B, AB, or O. And so if you are A, then your blood has the A antigen presented on it. If you are B blood type, then it means your red blood cell has these little proteins on the outside for the B. If you are AB, that means that you have both of them on there. And then if you are O, that means you have none of them. And so what happens is that your body also has this immune response. So they also have antibodies that recognize these antigens. So the way it works is that this little shape right here indicates that there's an antibody. And these particular antibodies recognize certain things. So the B antibodies recognize the B antigen, and the A antibodies recognize the A antigen. So kind of the shape is meant to distinguish this. So what you'll notice is if you're A blood type, you have antibodies that circulate in your blood that recognize the B antigen, but not the A antigen. And if you're the B blood type, you have antibodies that recognize the A antigen, but not the B antigen. And if you are AB, you don't have any of the antibodies. And then if you're O, then it means you have both of these antibodies. And so what happens is, like, this is a natural process where your body clears out all of the antibodies that recognize your blood. So if you donate blood, so if you are an O blood type, and you receive, for example, A blood, this is a big problem for you because you have these antibodies. And so if someone donates the A blood to you, these antibodies start going into overdrive and they recognize that blood type and they think this is a foreign substance. And so if you are an O blood type, that means that you can only receive O blood in a blood transfusion. Whereas if you're an AB blood type, you're actually pretty golden. So you don't have any sort of antibodies circulating in your blood plasma. And so if you have A go to you, you don't have a problem with it. B, you don't have a problem with it. O, you don't have a problem with it. AB, you don't have a problem with it because you no longer have the antibodies. And so this is kind of where the blood type came from, is this realization that, you know, that in a blood transfusion, there are incompatibilities between which blood you can receive and which blood you can give. And so the O blood type, that's the universal donor. That's the person, anyone can accept the O blood type because it doesn't have any antigen on it. But actually, if you're O blood type, you're kind of at a disadvantage in the sense that you can only receive O blood. All of the other blood would be really bad for you. So what is the basis of this? So there are the A and the B antigens. So these are a carbohydrate group bound to lipid groups on red blood cells. And there is also the H substance. So 1 or 2 terminal sugars added. So the H substance is kind of shown by these little lines right here. And then the antigens are carbohydrate groups that are added to the end of these things. Okay, so you'll see that they have these things added to the end. So there are 3 alleles of this gene that you have to worry about. There is the IA allele, there's the IB allele, and then there's the little I allele. So IA and IB, they produce the respective antigens. So these genes are necessary for creating these carbohydrate groups right here. So if you have the A allele, you are able to produce that A antigen. If you have the B allele, you're able to produce the B antigen. And if you have the little I, you're only able to produce this little stem right here, the H substance right here. So this is very interesting from a genetic perspective in terms of the relationships between that. And so we have 3 alleles. And so IA and IB are dominant to little I, but IA and IB are codominant to each other. So this are all of the 6 genotypes that you can have with respect to your blood type. So little I means that you're unable to produce any of the antigen. And so you have the O blood type. If you are heterozygote for A and B, then you produce both of the antigens. So this gene produces the A antigen and this produces the B antigen. And so this is where it's referred to as codominant because you're actually producing a blood group that has both the A and the B. If you're A blood type, you either are this genotype or this genotype, IA, and IA little I, and then IB or IB little I. So if you have, as long as you have one copy of the A allele, you're able to produce the A antigen. As long as you have one copy of the B allele, you're able to produce the B antigen. So again, these IB is dominant to little I, and IA is dominant to little I. So this is how blood type works, and now you can simply use kind of the Punnett square. You can figure out what alleles can be produced, what gametes can be produced, and you can use that to understand the inheritance of blood types. So part of the activity that you're going to do is to see how you can use that to exclude parents' paternity test in order to say whether or not a given offspring can have a given father or mother. All right, the final thing that we are going to cover for these alleles are lethal alleles. And so this is the situation where certain genotypes actually result in dead organisms. So this was discovered in agouti versus yellow coat mice. And so what they found was that if they intercrossed yellow mice together, they found a very unusual ratio. So typically we would expect to see a 3-to-1 ratio, but instead what they were finding was a 1-to-2 ratio. So instead of it being a 1-to-3 ratio, they were finding a 1-to-2 ratio. And so the reason for that is that there was a genotype that actually resulted in the mice dying in utero. So the mice were never born; they just died in the mother, and they were never seen. So how does this work? So let's say you have two alleles, and so big A results in a gouty colored coat, and if you're heterozygote, you have a yellow coat. And then if you have both homozygotes, you die as a mice. And so if we do a cross between yellow coat mice, this is the Punnett square that gets produced. Okay, so a quarter of them would expect it to be this, and then half you'd expect to be this, and a quarter that expected to this. However, because these mice never come out, we have to renormalize the probability. So this is very similar to what we did with the pedigree information. So all of the probabilities have to add up to a quarter. So typically you have 1/4 plus 1/4 plus 1/4 plus 1/4 equals 1, but now we only have 3 of those squares. And so if we add those all up, it's equal to 3/4. So the probability that it's agouti is 1/3, and the probability that it's yellow is 2/3, because we simply divide that by the 3/4. And so if you ever see a 1/3 to 2/3 ratio, immediately you should be thinking that this might be an example of a lethal gene.